This video is going to describe the functions of eukaryotic organelles. We'll begin with an animal cell. As you may recall, the entire cell is surrounded by a cell membrane, which is made up of the phospholipid bilayer. The organelles inside the cell are also biomembranes, which means that they are comprised of phospholipids with embedded proteins. Right now, we'll discuss each organelle and its specific function. The first organelle we're going to look at is the nucleus and the nucleolus, which is inside the nucleus. This structure right here is the nucleus. The nucleus contains DNA and also the nucleolus. The outside of the nucleus is made up of a phospholipid bilayer, a biomembrane of phospholipids and embedded proteins called the nuclear membrane. Inside the nucleus, you'll find the DNA. DNA in this picture is in chromatin form. So this is the chromatin right here. Chromatin is DNA organized around these proteins called histone proteins. So here's the DNA right here. It's coiled around the histone proteins to keep it organized and to keep certain pieces covered or uncovered, which we'll talk about later. So here's my DNA in blue, and then organizing the DNA in the center are these histone proteins in pink. Together, the DNA and the histone proteins are called chromatin. So chromatin is found inside the nuclear membrane in animal cells and in eukaryotic organisms in general. As I said before, the histone proteins help organize the DNA. Also inside the nucleus, we find the nucleolus. The nucleolus is a structure. It's kind of an informal structure where ribosomal subunits are made. So here we have the nucleolus. And the nucleolus will write the function underneath. The nucleolus makes ribosomal subunits. The DNA, during the process of transcription, makes mRNA, which is a copy of a specific gene in the DNA. So if this is the DNA, and this is a gene right here, DNA is transcribed into mRNA in the nucleus. Now, both the ribosomes and the mRNA need to exit the nucleus so that they can be used for protein synthesis. And the way they exit the nucleus is through holes in the nuclear membrane, and these holes are called nuclear pores. So the nucleus is the location of the DNA, where mRNA is made, and where the ribosomal subunits are made as well. Once the mRNA is made, it can be removed from the nucleus through the nuclear pores, and it can move on to the rough ER to help with protein synthesis. So let's move on to what the rough ER does. The rough ER, again, is a series of biomembranes made up of phospholipids and embedded proteins, and the rough ER is where proteins can be made into. It helps proteins fold. It's also involved in modifying proteins, adding carbohydrate groups or cutting proteins. So the rough ER does a couple things. It helps a polypeptide fold from the primary to the secondary to the tertiary structure. It can also modify a protein by adding a carbohydrate group to make a glycoprotein, or cutting a protein to activate it or inactivate it. This all occurs in the rough ER because, as you can see, and as we talked about earlier, the ribosomes are attached to the surface of the rough ER. These attached ribosomes are going to meet up with this mRNA that's left through the nuclear pores. And this mRNA is going to be read in the process of translation to make our polypeptide. So this attached ribosome is making a polypeptide into the middle of the rough ER. In the middle of the rough ER, this polypeptide can be folded up into its secondary and its tertiary structure, and carbohydrate groups can be attached to make it a glycoprotein. Other modifications can occur as well. It can be cut, all sorts of things can happen to it. Attached ribosomes make polypeptides into the rough ER, and these proteins are ultimately going to be used in the cell or on the cell membrane, or they're going to be exported from the cell. There are also ribosomes that are free-floating. The proteins that are made on free-floating ribosomes can only be used in the cell. They will stay in the cell. So now we have our protein that was folded up in the rough ER. And as I said, these are the jobs of the rough ER. This protein can then be transported to the Golgi. One way it can be transported to the Golgi is by using these transport vesicles. Transport vesicles are also made up of phospholipids. So this protein can be made in the rough ER. Part of the rough ER can pinch off and become a vesicle. And then these transport vesicles can transport this protein to the Golgi. Okay. They do that by moving along a system called the cytoskeleton inside the cell. So these little motor proteins will walk the vesicles to the Golgi. In the Golgi, more modifications can occur with our protein. So the Golgi has a couple functions. The Golgi, again, is a system of phospholipids. Once this protein arrives at the Golgi can modify the protein further, but most importantly, it tags the protein for transport, kind of like giving it a zip code. So the Golgi's job is to receive proteins, to modify proteins further, and to tag the proteins for transport. So it's almost like giving the protein a zip code. So let's say we want this protein to go to the cell membrane. Maybe the cell membrane zip code is 22030. We tag this little protein with the zip code 22030, and then that protein knows that it needs to be taken to the cell membrane to become a membrane protein. The Golgi has two sides, the cis side and the trans side. Now, sometimes we need to store proteins. We need to store other structures in the body. That's what the vacuole is used for. The vacuole is used for storage. Sometimes old proteins need to be broken down. That's the lysosome. The lysosome's job is to break down old cell parts. And because of that, it often contains digestive enzymes. The smooth ER is also on this big sheet. The smooth ER is again made up of a series of phospholipids. But while the rough ER is involved in the synthesis and modification of proteins, the smooth ER is involved in a couple things. It makes lipids. For example, your phospholipids are made in the smooth ER. It also, other lipids would be hormones. So it could make phospholipids, hormones, etc. It also detoxifies. In your liver cells, they have a really large smoothie R to help detoxify toxins. Also, your smoothie R can act as storage. For example, in your muscle cells, your smoothie R stores calcium, which helps with muscle contraction. Finally, in an animal cell, we have our mitochondria right here. The mitochondria is actually a double membrane. Okay? It has an outside membrane made up of phospholipids and an internal membrane as well. All of the surface area in the mitochondria is excellent because it provides a lot of surface area for reactions, which helps out with the efficiency of cellular respiration. Mitochondria's main job is to produce ATP through a process known as cellular respiration. This folded inner membrane brings back a concept we already talked about, which is the importance of a high surface area. This folded inner membrane helps increase surface area for reactions. Which means that the rate of respiration can be higher, which leads to an increase in the ATP production, which is very important because we go through ATP very rapidly to do active transport, to help cells grow and develop, to do all sorts of metabolic processes. The mitochondria is really cool because not only does it have an outer membrane and an inner membrane, it also has its own DNA and it even has its own ribosomes. And this double membrane, the fact that it has its own DNA and its own ribosomes, and it's kind of similar in size to a prokaryote, gives rise to evidence for the endosymbiotic theory, which we'll talk about a little bit later. Plant cells are also eukaryotic cells, so they have many of the same organelles that animal cells have. So if we do a quick labeling of our plant cells have a nucleus, which is an organelle surrounded by a phospholipid bilayer with DNA and the nucleolus inside. They have ER. They have mitochondria. They have Golgi. They have the cytoplasm. They have ribosomes. They have an internal cell membrane. Well, internal in that it's inside their cell wall, but it's still the cell membrane. And then they have all the internal membranes, the nuclear membrane, the ER, the Golgi, etc. They also have a cell wall. In plants, the cell wall is a structure that is made of cellulose and again, it helps provide structural support for the plant cell. It's an adaptation that allows plant cells to exist and even thrive in hypotonic solutions, whereas animal cells placed in hypotonic solutions lyse because they don't have this firm cell wall. They also have the chloroplast. Here's the chloroplast right here. The chloroplast is able to harness the energy from the sun using chlorophyll and other pigments. It can harness the energy from the sun to do photosynthesis. Photosynthesis converts carbon dioxide and water into glucose, into sugar. This is also a product of endosymbiosis, which we'll talk about later. So plant cells have both the mitochondria that does cellular respiration and the chloroplast that does photosynthesis. Finally, a major difference between plant and animal cells is this large central vacuole. Animal cells might have smaller vacuoles for storage, but only plant cells have this large central vacuole. It stores water and helps maintain turgor pressure. It can also store pigments. And again, this turgor pressure puts pressure on the cell wall to help with rigidity. Remember, plants don't have skeletons. This large central vacuole actually increases the surface area to volume ratio of a plant cell. And plant cells are often larger than animal cells because this large central vacuole is not something that the plant cell really needs to take care of in terms of like maintaining homeostasis, as it does for like the other more energetically needy organelles. That's why plant cells can be larger. So one thing that it would be good to do for you is to review animal cells versus plant cells. Those are the two main types of eukaryotic cells we will be studying, but please keep in mind that protists and fungi are also eukaryotes. Later on, we'll look at another type of cell, which is a prokaryotic cell.